depending on each person to keep the time on time. I'm going to start first. And I'm coming from Japan, and this is the part of my profession of the paintings. This is the apple trees in suburb of Sapporo. I painted last year for the. It's a nice, beautiful city in Sapporo. Will be held in the near future in the Winter Olympics back again. But my title is Modulation of the Status of Influenza Infection by AKT Peptide and Akt-in. This is the little bit complex slide, but I'm more focusing on working on this intercellular signal transduction through the PI3 kinase AKT pathway. This pathway has been characterized past 2 decades or so, and mainly I think it's focusing for the anti-apoptotic action through the some numbers of the anti-apoptotic pro-apoptotic molecules phosphorylation. In addition, these molecules control the cell cycle or glucose metabolism, protein synthesis, angiogenesis. So these molecules of serine kinase controls wide variety of the cellular events. So in past 10 years or so, PI3 kinase AKT signal and the viral protein has been shown to be interact. For example, EBV virus protein LMP1 or P2 interact with the PI3 kinase component P85, or the HCV or HPV for the hepatitis virus particles HCV N85 or the HBX interact with the other component of the PI3 kinase P110 or P185. And the same is true for the HIV for the AIDS virus. And those are interacted and activated, and perhaps I think it's preload for the chronic infection or the latent infection, and some of the events for the tumorigenesis or cellular transformation. And in addition, there's an acute infections phase is also modulated, has been demonstrated by RSV or the influenza. We are talking about the NS1 AKT interaction through the acute phase of the influenza infection today. So this is the replication cycle of the influenza virus, as most of the people are aware of that. The influenza virus get into the cells and the transcribe, but because this is the RNA virus, so most of the component transcribed and replicate and propagate inside the cell is utilize the host cell factors, and then replicate it and comes back again and excitosed and comes to out of the cells for the replication. So in this process inside the cells, the viral particles utilize some numbers of the host cells. So there's a whole lot of things are going on inside the cells. One of the things that we are focusing on is the PI3 kinase AKT pathway and the interaction perhaps for the influenza virus. And with these assumptions, we may be able to modulate the inhibition of AKT will have an impact for the infection or the replication stage of influenza virus infection. So this is a structure, and as there's 8 proteins are coded by the RNA, and we are checking all the component of the influenza virus because the AKT has been shown to be phosphorylate its substrate as the sequence of RXR, XXST. This is the serine residues phosphorylated by the AKT. So we analyze all the component of influenza virus, and what we came across was the NS1 non-structural protein 1 of the influenza virus. This is consisted with the 230 amino acid and 26 kilodalton, and the 2 binding motifs, one for RNA and one for the effector domains. And this protein is also mutation of this has a potentiate viral toxicity. So this has a prior for the infectious stage of the influenza virus. So first we undertook the conventional standard co-immunoprecipitation assays to overexpress both of the flag and HA tagged NS1 and AKT into the 293 cells, and this indeed interact in the mammalian cell in the overexpression systems. And next we conducted using the PR8 as an experimental sort of infection into the A549 cells. And indeed, this infected cell is also interacted with the AKT of the host cell and the viral protein the NS1 in the infected stage. And this interaction is indeed functional. This is the in vitro kinase assays. So this increased amount of NS1 correlate well with the phosphorylation of AKT, which is 473 of serine and 308 of the threonine, in addition to its substrate of GSK, which is also important for the replication, has been shown to be important for the replication and entry of the viral particle. So this interaction itself is physically interact and has some function. And that is in the test tube experiment, and this is in a more cellular context, and with the stimulation PDGF, then the AKT is phosphorylated, and which is correlated well with the NS1. So inside the cells with the increased amount of AKT phosphorylation also phosphorylate NS1. And we undertook the LLC mass analysis to identify where the phosphorylation site, and it is indeed threonine 215 is the locus of the AKT to phosphorylate in this NS1 protein. So this assumption we sort of proved, and this is sort of the models. The viral particles provide the NS1 to interact with the AKT for provide the anti-apoptotic actions, and the protect the cells for more beneficial for the propagations, for some symbiotic relationship. So the next step is when if we block this step, then what happened for the viral infection stage. So this is another paintings on the suburb of the Brewer factory close to Sapporo. So this is structure of TCL1 we characterized a while ago. And this molecule, TCL1, the T-cell leukemia 1 of the leukemia proto-oncogene. And this is shown to be interact with AKT, and this beta E and beta E seed is the interface for the to interact the plexurin homology domain of AKT. We through the numbers of functional and the analysis, we identified this beta A, half of the binding sheet has the ability to inhibit the AKT. So this is the amino acid alignment of, there are 3 families of TCL1, and this is the locus where we identified has some ability to bound and inhibit the AKT kinase activity, as I show you in the next couple of slides. So these are reasonably conserved among these families. And this is the sequence, basically a 15 amino acid, and there's some conserved motif right in the middle of the beta sheet. So this is again the in vitro kinase assay of the test tube experiment, and increasing amount of either TCL1 sort of amino acid based Akt-in or the TCL1 based Akt-in inhibit the AKT kinase activity and along with its substrate pretty well. And the same is true for the cellular context. We introduced this peptide of TCL1B based peptide in this case, and stimulate with the PDGF to activate AKT, and compared to the mock transfection, mock transduced cells, TCL1 based Akt-in transduced cell compromised the AKT kinase activity shown by the threonine 308, and even after the PDGF stimulation over here. And what happened for in the cellular context for the replication? So we compare as a comparison, we use the LY294002 as a PI3 conventional PI3 kinase inhibitor, which inhibits the AKT kinase upstream molecules as well as the NS1 phosphorylation. And with the treatment of either TCL1, TCL1-based Akt-in also inhibit the AKT, this is control. So compared to that, it's a compromised phosphorylation, as well, which is correlated well with the phosphorylation levels of NS1. And this constantly, this is the colony assays, and either the PI3 kinase inhibitor or the TCL1, TCL1-based Akt-in also inhibit the replication of the influenza in this experiment. So this indeed is the shown that the dose escalation of either TCL1 or TCL1-based Akt-in compromise either the NS1 or the AKT kinase activity in a dose-dependent manner over here. And the same is true for the TCL1B, the NS1 phosphorylation along with the phosphorylation of AKT is correlated well for the declining its activity. So this is the replication of the colony assays, or the increasing amount of TCL1 or TCL1B declining its ability for the replication in a dose-dependent manner. And same as true for the, we check the time dependency with the after the 24 and 48 and 72 hours later, the phosphorylation levels NS1 is declining time-dependent manner, along with the phosphorylation levels of AKT over here. And the GSK3, as I mentioned, this is important factor for the early entry and the replication of the influenza virus, has been shown in the literature. So this is the quantitation of the graphs on 24 to 48 and 72 hours. Those peptide inhibitor compromised its replication in a time-dependent manner. Next, we investigate how that happens, and we check examine the levels of the two of the inflammatory cytokine, in this case IL-6 and IL-8. And we are sort of surprised that if we treat the cells with the LY294002, which is a PI3 kinase inhibitor, it inhibit the production by IL-6 or the IL-8. But the TCL1, TCL1B-based Akt-in, although this has the inhibit the replication or the entry of the virus, but it does not influence any of the impact on the inhibition for the inflammatory cytokine of neither of these IL-6 or IL-8 over here. So then we move on to check the confocal microscopy to study the presence or entry of the viral particle into the cells. So we use the nucleoprotein as a marker, existence marker for the influenza, and the co-immunostained with the phosphorus level of AKT phosphorylation. So the control treated cells, either here or over here for the control peptide, show the presence of the influenza along with the phosphorylation levels of AKT over here, and they're shown as yellow. However, if we treat the cells either the LY or the TCL1-based Akt-in, then the green color and the yellowish color is gone away, which indicate the influenza virus is no longer get into the cells and activate the AKT and within the cellular context. And this is the quantitation of the previous slides. You can see either the nucleoprotein, which is the presence of the or entry of the influenza virus, is inhibited with the TCL1, TCL1-based Akt-in, and the same is true for the phosphorylation levels of AKT is compromised. And it's comparable to the conventional PI3 kinase inhibitor LY294002. So this is the sort of the summary we undertook in the shown over here. With the inhibition, since the viral particles utilize the numbers of the host cell molecules for the replication or entry, and we use the one of the crucial factor for AKT to inhibit by the our peptide Akt-in, and which presumably inhibit either the early stage of viral entry and the replication, and the modulate the infectious stage of influenza. So this is the summary, which basically we show that this T-serum-based Akt-in suppressed kinase activity and the levels of the influenza virus phosphorylation. And this inhibition of activity suppressed viral replication and dose and time-dependent manner. And this Akt-in viral replication without inhibiting the production of inflammatory cytokines such as the IL-6 or IL-8. And therefore, the inhibition of AKT kinase activity in the host cell or the virus entry or the early stage of viral replication, and possibly through the inhibition of GS3K beta, which is AKT substrate. So these are the people involved in the work, and this is from my group in the current institute, and the structure study are done when I was engaged in the Boston, and these are structure study are done by the Christian Lomenstadt in the Montpellier, and this is another view, downtown view of the Sapporo Suzuki no downtown. It's a very nice cozy time, packed with snow during the winter. Thank you for your attention.
Thank you very much, Dr. Noguchi.